Bismillahirrahmanirrahim. Let's start the topic acute inflammation. Acute inflammatory response rapidly delivers leukocytes and plasma proteins to the sites of injury. Once leukocytes are there, they clear the invaders, start digesting and getting rid of the necrotic tissue. Acute inflammation is having two major components. The first one is called vascular change, that is vasodilation, and changes in the vessel wall that will permit the plasma protein to leave the circulation, and it is called increased vascular permeability. In addition, endothelial cells are activated, resulting in increased addition of leukocytes and migration of the leukocyte through the vessel wall. Cellular events include the immigration and accumulation of leukocytes from the leukocytes at the focus of injury. This is called cellular recruitment, and it is followed by activation of the leukocyte. As a result, they are enabled to eliminate the offending agent. You can see over here I've written that the principal leukocytes are neutrophils. Let's talk about the stimuli for acute inflammation. It includes infections. It can be bacterial, viral, fungal, parasitic. Trauma due to any blunt or penetrating object. And various physical and chemical agents, for example, thermal injuries such as burns, radiations, toxicity from certain environmental chemicals. Tissue necrosis, it can be from any cancer, including the ischemia, as in myocardial infarct and physical and chemical injury. Foreign bodies, for example, sutures, crystal deposits, splinters. Immune reactions, also called as hypersensitivity reactions, against the environmental substances or against the self tissues. Because the stimuli for these inflammatory responses often cannot be eliminated or avoided, such reactions tend to persist with features of chronic inflammation, and this is called immune-mediated inflammatory disease. This is a picture about the vascular and cellular events of acute inflammation. The major local manifestation of acute inflammation compared with normal. The first one is the vascular dilation and increased blood flow. It will lead to erythema and warmth. Second one is extravasation of plasma, fluid and proteins that will lead to edema. And the third one you can see, that is neutrophil immigration. That is leukocyte mainly neutrophils. Immigration is shown leading to accumulation. So that's all. Let's talk about the recognition of microbes, necrotic cells and foreign substances. First, let's talk about the pattern recognition receptors. They are the receptors that recognize structures that are common to many microbes or dead cells. Phagocytes, dendritic cells, and many other cells such as epithelial cell express receptors that are designed to sense the presence of infectious pathogens and substances released from dead cells. Toll-like receptor and inflammasome are the two most important families of these receptors. Toll-like receptors are the microbial sensors. They recognize the product of bacteria, virus, and other pathogens. They are located in plasma membrane and endosomes. Recognition of microbes, active transcription factors that will stimulate the production of number of secreted and membrane proteins. Inflammasome is a multi-protein cytoplasmic complex that recognizes the product of dead cells such as uric acid and extracellular ATP, as well as crystals and some microbial products. Triggering of the inflammasome results in activation of an enzyme called as casapase 1, which cleaves the precursor's form of inflammatory cytokine interleukin 1 beta into its biologically active form. Interleukin 1 is an important mediator of leukocyte recruitment in the active inflammatory response, and the leukocytes phagocytose and destroy dead cells. Over here I have written the joint disease gout. It is also caused by the deposition of urate crystals, which are ingested by phagocytes. When they are ingested, they will activate the inflammasome. As a result, there will be interleukin 1 production and acute inflammation will be there. In atherosclerosis, that is associated with deposition of cholesterol crystals in vessel wall, and obesity associated type 2 diabetes. interleukin-1 plays an important role in causing these diseases. This finding raises the possibility of treating these diseases by blocking interleukin-1. This is the diagram. Over here you can see the toll-like receptors that are located in plasma membrane and endosomes and other cytoplasmic and plasma membrane receptors. They recognize the product of different class of microbes. The proteins produced by TLR activation have numerous functions, but right now we only know its role in inflammation. Then inflammasome is there. You can see pathogenic bacteria extracellular ATP is there. Inflammasome consists of a sensor protein, an adopter, and enzyme caspase 1, which is converted from an inactive to active form. And then you can see they are playing their role in acute inflammation. Toll-like receptors, along with inflammasome, they are going to play their role in acute inflammation, and that is how there was recognition of microbes, necrotic cells, and foreign substances, activation of these two receptors, and their response towards acute inflammation. That's all.